I try to answer questions people have about trees and forests, and I think we have one tonight, so
yeah, we do.
hopefully I'll be chiming in.
That's great. I don't mean to put Karen on the spot, but since you're here, we maybe won't have a question for you, but you do work for the Harris Center. Why don't you tell us a little bit about yourself?
Sure. My name's Karen Rent. I'm a teacher naturalist. I work with mostly elementary school kids. I'm not an expert in anything, but I love learning and I know little bits about lots of things. And if you have any questions about games that teach ecological concepts to children, I could answer that for you.
I love it. Thanks, Karen. And how about John? How about you?
Hi, everyone. I'm John Benjamin. I tend to answer questions about fungi and mushrooms at these gatherings. So I think I'll be having a couple questions tonight about that. So.
It's great. And John's also great at answering some snake questions when we have those. And tonight, this is so exciting. We have a special guest who is here, who we asked to come, and he said yes, and we're thrilled. So Mark, why don't you tell us about yourself? Oh, you're muted. Got to unmute yourself.
Rookies, right? Yeah. Thank you very much for the invitation. I'm glad to be here. My name is Mark Ellingwood. I live here in Hancock with my wife, Susan. We're members of the Harris Center, have been for many years. I'm a chief of wildlife for New Hampshire Fish and Game. I've worked for 40 years in the wildlife profession, and jack of all trades, perhaps ace of none, but I'm delighted to be here and look forward to chatting with you about moose.
Oh man, Mark, I'm so glad you're here. It's awesome. And I love whenever Mark has done programs for us, I love that he doesn't just have knowledge, that's amazing, but a great sense of humor. Always appreciate that. So I noticed that our insect expert is not quite here, and I know she was going to be a little bit late, but we will be joined by Jenna Spear O'Mara, and she is an entomologist, and we do have quite a few insect questions. So hopefully she'll show up before we get our first question. So Miles, tonight we are starting off with something exciting.
Yes, we want to get an idea of our audience here, and we want to find out how often you're visiting Harris Center land. So I'm going to launch a poll, and you guys can participate on your screens here, and we'll find out what you guys are up to. We will be doing 2 other polls during the presentation and seeing what you guys think of our mysteries tonight.
It's exciting. Oh, the technology we have.
All right. Well, we have most people responding, and I'll go ahead and share the results. So we have a lot of people visiting, you know, more than once, but some people have never been.
That's great. Well, we are a Zoom audience, so we might have some people from very far away. So if you ever do make it to Hancock, make sure you stop in and check out the Harris Center. We'd love to see you. All right, shall we go to our opening slide?
Let's do it.
All right. Okay, fabulous. So this was the question that drew me to ask Mark here. We have a moose question, and I know that Mark, for many years of his work at Fish and Game, has been involved with the moose population. So Mark, what is the health status of moose with their plague of deer ticks, signed Betsy? So tell us what you think, Mark.
Okay, Betsy, thanks for the question. Look forward to sharing some information with you. The issue of moose is actually related to winter tick, a different species of tick than the deer tick. The difference between those 2 species is that the moose tick has a 1-year life cycle versus the 2-year life cycle of deer ticks. And of course, they're different visually as well. They have different anatomical features, but we're talking about winter ticks when we're talking about impacts on moose. Winter tick actually exists throughout the state, but is most notable where we have our highest densities of moose. So a couple things for you to understand relative to the impacts of ticks on moose is that in large part it's driven by density. So the more prey species you have, the more potential for impact, the better the tick population thrives, and the greater impact that tick population has on those moose. A couple other things to consider is that there are other important mortality factors on moose aside from ticks. Down here in southern New Hampshire, brainworm, which is a parasite common to deer, benign to deer, but fatal to moose, is likely a bigger impactor on our moose population south of the Whites than actual winter tick. All that said, just give you a little background. As of the mid-2000s, at or around 2007, we had something on the order of 7,500 moose in the state. Today, we have something closer to 3,000.
Wow.
And that reduction has been driven by moose, by winter tick, in mainly northern New Hampshire. The impact of ticks on moose is highly variable throughout the state. So you shouldn't necessarily assume that the impacts that are occurring in the North Country are the same as those occurring here. In fact, we know that's not the case. And we consider brainworm, deer parasite, to be a bigger impactor on moose down here than the ticks themselves. In any case, little background regarding the impact of winter tick on moose. You can have upwards of 30 to 70,000 ticks per moose during a bad year in northern New Hampshire. And most of the comments I'll make are specific to northern New Hampshire. Those ticks are picked up in the fall. And depending on time of first snow, the tick loads on moose can be either heavy, moderate, or light. If you get an early snow, then those ticks, which are out in our woodlands trying to hitch a ride on moose, are losing their ability to catch a ride. And as a consequence, early snows means less ticks, shorter winters means more ticks, warmer climate means more ticks, and more ticks means less moose.
Wow.
In our case, what we discovered beginning in the early 2000s that on occasion, tick numbers would go through the roof. And we were describing what we call an epizootic event where we would lose 50 to 75% of our calf crop as a result of that increase in tick numbers. Those were infrequent events at the time, once every 5 years, something on that, or perhaps less. More recently, continuing that research from 2014 through the present, those epizootics, or those increases in tick numbers, are approaching 3 and 4 out of every 5 years, and we're losing upwards of 60 and 70% of our calf crop during those events, and the population cannot sustain itself under that level of mortality. What happens is the ticks pick up a ride in the fall, they mature, they're on the ticks in January, February, and March, they take a blood meal. And if you have enough ticks on you, you can literally recycle the entire blood volume of a moose in 2 weeks. The animal can't sustain that kind of loss of protein and nutrients, and as a consequence, the youngest, smallest, weakest of animals in the population suffer the most, and that suffering is handled by our calf crop. And so we're referring to those as calves. They're really 10 months or so old, but their size is such that they're very vulnerable to heavy predation, and we lose that impact. Across the state right now, we have something on the order of half of the number of moose that we had 10 and 15 years ago, but those numbers appear more sustainable. Down here in southwest New Hampshire, the moose population has ebbed up and down at modest levels, something on the order of an 1/8 or a 1/10 of the northern population. It's always been that distinctly different. But our population in the last several years has actually grown slightly. So right now we have something on the order of a moose every 4 square miles in southern New Hampshire. We have upwards of 2 or 3 moose per square mile up north, where they're suffering from the greatest impact of ticks. Something that's interesting that Maine is doing right now, they have the advantage of dramatic array of latitudes. They're experimenting with their ability to control ticks by controlling moose density. So the irony is, if you want healthier moose, you may well have to reduce moose populations below that threshold that favors abundant tick presence. So there's some basics. I could talk about this for hours. I won't do that to you. But if anyone has any related questions, I'd be glad to answer them.
Here's a related question from Kim. She writes, I've heard that moose are moving further south than they used to be. As they do, will these parasites come with them?
The parasites are here already. The abundance is driven by the prey base, which we understand and have experienced to be moose themselves. Moose are particularly vulnerable to ticks because they don't self-groom. They didn't evolve in the presence of ticks, and so they haven't adapted to that presence. They don't remove ticks efficiently as compared to something like white-tailed deer.
Wow. And just briefly, Mark, you said that brainworm in this part of the state is a bigger problem, and that's a parasite. Can you just talk a little bit real quickly about that life cycle? I know it has to do with deer poop?
Yep.
Tell us about that, because poop is my thing.
Okay, good, and I'm counting on you to cover the poop questions. Deer have brainworm. It's present in various levels of deer, in some part dependent on the density. The animal, when it defecates, it drops the cysts or the eggs for that nematode that then prospers. A snail picks it up, and the snails are inadvertently consumed by moose during browsing. Once digested, the nematode penetrates the digestive system, looks for a nerve wherever that may be in the body, follows it to the spinal column, travels up to the meninges of the brain, and it results in the death of the moose.
Man.
So we've done some interesting research recently with the University of Tennessee or Tennessee Tech, where we sponsored with other states in New England the development of a blood test that will allow us to determine from a blood sample, whether it's a road-killed moose or a hunter-killed deer, what the prevalence of brainworm is in our populations. One of the ironies of this entire thing is both as ticks and moose impacts are driven by a warming climate, deer densities are increasing as a result of milder winters. We have less deer yarding. The higher the deer density, the more the impact of brainworm on moose. So all these things are connected.
Wow, fascinating. Thank you so much, Mark. I'm just so happy you were here to take this question and you gave such a great, thorough answer. Awesome. So we are going to move on to the next question. And this, am I right? Is this a poll question, Miles? So we're wondering if, let's just go back to the slide for a second so I can read it. So 2 scat sites, same animal. Source of the scat, this was on a path in the woods in Alsted in July. The card key gives an idea of the size, big, and it isn't tapered, which would indicate canine from what I've learned from previous sessions. Good job, Tricia. She's been paying attention to my comments about twisty canines tapering scat. So let's see what the poll, what do we got here? We have some choices for you. Do we have the poll up? Is it back up?
Can you guys see
Nope.
it? Got some answers coming in. It could be just behind your presentation window.
All right.
I can launch it again, I think. Hold on. Relaunch. Here we go.
There goes the poll. So whose scat is that? Coyote, bobcat, black bear, moose, or other? That's good. Getting some more votes coming in.
Susie, it's very lined up neatly next to the car key.
Yeah, it's really good. Love it. So, Miles, what's our results? Black bear is the winner at 67%, and that is actually correct. Let's take a look at that scat and kind of talk a little bit about it. This is a little gross. I hope nobody's eating any snacks with their beverage this evening, but bear scat kind of looks like human scat, and it's sort of rounded on the ends and a little segmented. It's not tapered. It's often made up of a variety of materials because they are very omnivorous. And in this, you might even see sort of what I'm noticing is shiny bits, which would mean to me that this bear was feasting on some perhaps insect larvae and then some seeds. I see some sort of seedy pieces. And it's kind of not one theme. A coyote scat is often one theme. You see a lot of fur and bones in coyote scat, but in bear scat, it's often mixed material unless they've been gorging, which is now beginning on the fruits of the season, like apples or peaches or berries, and then it will be very large and have the majority of material in there from that one source. Mark, do you have anything you want to add about bears and bear scat?
I would just say it's highly variable across the year. Some people perceive those fall fruit scats to be something they refer to somewhat crudely as bear puke. And it's prevalent and widespread in orchards to the extent where you have to walk where you step, but continues to be my belief that those are actually scats from the animal, and the fermenting and the overabundance of fruit in the digestive system prompts some very exciting action.
Very exciting action. Bears are moving into the hyperphagia stage of their life right now, where they're just eating as much as they can during the feast of late summer into fall to try to pack on the weight. And so they're really like overeating. And you know what happens when you overeat? Overaction in the gut. So there was a bear related question that I wanted to ask, and this is actually from a neighbor of yours. And this is the question. Let me just read it to you. This person, Pierce, thinks that he has a bear digging up yellow jacket larvae near my peach trees. And what any things that might prevent that or stop that from happening? I'm looking at Mark to see if you have any comments about that.
First of all, I'll mention the neighbor across the street from Pierce, I happen to know, lost quite a few chickens recently. And just a quick note, if you want to have chickens in the state of New Hampshire, you need electric fencing or you're going to lose them to bears. Bears love chickens. They like pigs as well. So those are species that really need protection. My guess is that Pierce may be seeing skunk activity rather than bear activity. You know, bears are quite a presence. That would be substantial digging. But Pierce is correct. Bears love larvae and they dig wasp and hornet nets whenever they can. It's a favored food. They're adapted to doing that. And so it's possible, but in a lawn scenario, I'm certainly seeing skunk activity here in my lawn right now. And perhaps that's skunk. But in either case, both species are very interested in the larvae of those ground nesting hornets. And so this having been a dry year, hornets are very abundant in the ground. And so those animals will exploit them. In terms of turning them away, honestly, nothing short of electric fencing is going to effectively manage the movements of bears, but they are very sensitive to electricity. I'd tell Pierce probably just best to wait it out. Again, I suspect it's skunk rather than bear, but if it is bear, they're very focused. They'll eliminate those nests in short order, and that activity will come to pass quickly, I'm sure.
Great, thank you, Mark. That was awesome. All right, let's see what our next slide is. Let's see. What are these and are they a problem? I've never seen these before this year, but this summer they are popping up in our mulch frequently. Signed, Anne. This looks like a good question for our fungi, John Benjamin. Fungi man, what's up? What are these things?
Well, a great question, Susie. These sets of pictures were really interesting, and I have a couple of strong theories. I'm not totally sure, though, to be honest, based on the pictures, but it's always a fun thing with mushroom photos or fungi photos, because often you really need to get in and look at details. If you have a couple photos from afar, it doesn't often give you the details you need to really determine what's going on, per se. But I have a couple of theories, and I'm pretty sure I think I know what's going on, generally speaking. So one of the key factors of any kind of fungi ID is the substrate and the environment, like what's going on, where do you find it? Because, some mushrooms grow only in the presence of certain associative symbiotic trees that they need to have, or, certain kinds of, saprophytic decomposition situations. So it's all different. But the fact that it's mulch really limits it right there, because there are certain things that are very common in garden mulches. And actually, it's pretty interesting that a lot of things have been propagated by dissemination of mulch for gardens, like certain fungus species, including, I learned from a book recently, a lot of psychoactive psilocybin species have actually been propagated. This is not related to this picture at all, but it's very interesting, the things that have been selected for by our own practices as humans, how we mulch things, and then it gives saprophytic species an advantage in these very, very disturbed habitats in gardens and, schoolyards and whatever. Anyway, so what's interesting about these photos is that kind of, my first thought is it's a slime mold, which is not even a fungus. So a slime mold, this is a whole weird world of life, but I think this is probably Papilio. Okay, hold on. What's the name again? It's the dog vomit slime mold. I know you all wanted to know that. It's Fuligo septica, I'm pretty sure, which is the most common slime mold to find in your gardens. And when you find it at a certain phase of its existence, it's bright yellow, just unmistakable, very, very noticeable. But a slime mold is very strange. They're the Myxomycota. They lump them in with fungus because they don't know where else to put them, but they're totally unique with their evolutionary history. And, long story short, they start their lives as little individual cells, and they conglomerate over the course of their life cycle. And then their little membranes get merged and become a multinucleate plasmodium. And then they move around looking for nutrients. And then they can really solve problems and move quickly. You can actually observe them moving over the course of a day, which is kind of unique, and there's a documentary, I can't remember the name of it right now. It's all about how they've used slime molds to try to, they can solve problems about the most effective ways to navigate between one place to another. So they gave them a grid based on the Tokyo subway system, and they put little oatmeal slices on the points of the hubs, and they solved the grid really, really fast, way better than humans could have done as far as what best connections to make between these different hubs. So it's a very cool organism. I won't go on about them too long, but I'm pretty sure that's what that is. But also it kind of looks like it's getting nutrients from an old mushroom, which is a common thing to see. You'll find some types of fungi or slime molds digesting old mushrooms because they're ephemeral. Their only job is to send out spores, so they become food for other things very quickly. So what I kind of think you have here is a mushroom that did its thing, and then a slime mold found it as a nice source of nutrients to just kind of do its thing. And it's in the process of, when it becomes visible, it's in the reproductive phase. So it's sending out spores, basically. And, like all mushrooms do, but, slime molds, even though they're not mushrooms, do the same thing.
Wow, John, that is really outrageous information. Love it. And John and I were just connecting over a book we both read this summer called Entangled Life by Merlin Sheldrake. And if you want to read more about the fascinating world of fungus, we both highly recommend that, right, John? That book was.
Yes, indeed.
Incredible.
Yeah.
Incredible. All right. All right. I just love this picture and I love this description. This is from Max and Connie, who I say hi to if they're here tonight. Have had this lovely bracelet for years. Can you please identify the artist? I just love that. It is a bracelet of scat and it is so delicate looking. And yes, this is actually the connected scat of a porcupine, probably a young porcupine. And the reason it's connected is probably eating grass. So that's why it didn't really chew up all its pieces that well. And so when it defecated, it came out connected. And that's pretty common that you might find something like that in the springtime when they switch from eating the inner bark of trees of winter and move on to the meadows and are eating the grasses. And they're kind of like me after winter. I just can't wait to eat fresh greens. And then you maybe eat a little too much and you eat it a little too fast. And then this is what happens if you're a porcupine. You leave a beautiful little necklace here. So that's my theory on this lovely bracelet. So thank you, Max and Connie. And we'll head right to the next slide. What would have stripped these trees like this? Looking forward to hearing. And what can you tell me? And this is from Sharon Smith. And I'm wondering if Jenna has appeared in our...
I have.
Oh, Jenna. Oh,
yeah.
There you go. So Jenna and Jeremy, because this is also involves trees. So Jenna, what do you got to say about this?
Well, to my eye, and this is, I haven't been to this location, but to me, these look like trees that were not doing well. And then some insects moved in. So typically in our area, beetles and carpenter ants and termites and things will move into a tree that's ailing and not doing so well. If you're in the West, a lot of times bark beetles and wood boring beetles will attack a healthy tree. But in the Northeast, most of the time, trees that are not doing so well will be targets for a lot of wood boring or wood feeding insects. So I think that's what happened here. And then the bark sort of sloughed off. I don't think that any animal, large mammal or anything was taking the bark off. I think this was just a natural sloughing off of bark, but I would be interested to hear what Jeremy thinks.
Yeah, well, Jenna, I agree completely with you. I don't see teeth marks or scratch marks or any of the other signs that some kind of mammal or a bird has been actively working on these sites. I do, if we go back to that first slide, I do think there's something interesting here where these are white pines, I think. It's a little distant from my eyes, but I think they're white pines, and as the bark sloughs off, it will slough off in large plates. But look how the trees are, between them is what looks like a walking trail. And so my guess is that, yes, the bark has been sloughing off naturally, but it's been helped by some eager fingers of passerbys prying up the edges of the sloughing off bark and tearing it off. So that's why it's so perfectly taken off on the tree on the right and also on the left. And that's just my guess on that. But it's a natural process. As a tree dies, the bark will slowly slough off and accumulate at the base of the tree, and the tree can stand for many more years, even decades, as a bare snag without bark on it. But yeah, there's been lots and lots of animal activity in terms of beetles in the death of those trees and even after the trees were dead. Do you see any? I don't really see any powder post beetle activity in that, but there could even be something like that once the tree's dead going on.
Great.
Yeah, once the trees are dead, the beetles and things can really attack it well because they're not going to get the tree defenses pushing them out with the pitch or sap. So it's a lot easier for the insects to really get they can really move in there after the tree is dead, or at least when it's very ailing, very old, etc.
One thing to keep in mind is anytime you see a trail, it means that there are a lot of humans around, and humans do really odd things in the woods, and so they can create situations where you're like, what's going on here? And it's actually just the fact that there's human behavior around. I call it the Boy Scout syndrome, where if you think about young it doesn't have to be boys, it can be anyone, but I remember, whacking off branches with a stick and then the tree looked like it was pruned, but it had never been pruned, and all kinds of things like that.
Thank you. That's good. I like that. Very good. Oh, this next slide. We took these photos at Norway Pond. Could someone please talk about these freshwater mussels that the kids so love to pull out of the pond? Signed, Kathy. I will take this because I live right near Norway Pond, and for many, many years with my children, they have engaged in what Norway Pond kids do. They collect the mussels and kind of make piles of them and then skip them back into the water. And so I got really interested on who are these mussels and what are they all about? So the mussel that we're seeing at Norway Pond is actually the most common freshwater mussel in New England and probably in the Northeast. It's the common Eastern Elliptio. And this is a very common one found throughout our areas in ponds, lakes, streams, and rivers. And what's so cool about mussels, I kind of fell in love with mussels after reading this incredible book called Immersion by Abby Gashko. I think that's her last name. And it really made me see how important mussels are. They're kind of the keystone to the water, that fresh water. They are filter feeders. And so they're filtering the water consistently. And that means they're filtering out a lot of pollutants and they're filtering out a lot of bacteria. And they're really essential. And not only are they filtering, they're also an important food source for things like otters and raccoons and muskrats. And these mussels are actually, freshwater mussels are the most endangered invertebrate in North America. Not these in particular. The Eastern Elliptio is one of the most common ones, and its status under, concern is no concern at all. These are sturdy mussels that seem to be able to withstand a lot of abuse of kids skipping them and a lot of kind of pollution in their water. But without them, our waters would be a lot more dirty. And just such a cool thing about mussels, which some of you might remember, but mussels have this amazing symbiotic relationship with fish, where once the eggs are released into the water and the babies hatch, the babies called glochidia attach themselves to the fins and gills and scales of fish. And this particular mussel is a specialist in perch, both white and yellow, pickerel, smallmouth and largemouth bass, just to name a few. And then they mature, they kind of get encapsulated in the fish on the gills or in the scales, and they don't do any harm to the fish. And then once they become a juvenile, which is a very small, the tiniest little mussel you ever saw, they'll drop off the fish and land in the sand where they'll mature and grow. And that's where they'll spend decades. It's estimated that mussels like this can live for decades in the water. And I just kind of am in love with mussels. And whenever I'm at the beach with the kids, I'm always telling them about the mussels. They probably think I'm a crazy mussel lady. And even though they like to kind of collect them, most of the kids now make sure they keep them in the water so they stay alive. So that's what I have to say about that type of mussel, which is pretty cool.
Hey, Susie, how quickly we see trails from them moving? How quickly are they moving along the line?
They're not fast movers, as you can imagine, because they're a mollusk and they just kind of have that kind of foot or that particle piece that sticks out. So what I have noticed is kids will collect the mussels or kind of have them on the shore, and then in the morning when you come back, when I walk my dog down by there, they'll have moved, maybe 2 feet out, and then they'll kind of keep moving, and their whole cycle is pretty amazing. They just kind of stick in the ground and they open up their shell, and there's a series of fine filters that collect bacteria, protozoa, algae, and it goes into their kind of mouth and it gets sorted, and anything that they're going to eat kind of goes into their system, and everything else kind of gets passed through them, and that's what helps filter the water, which is so cool. And I should mention that mussels for a long time were an edible food source for the indigenous people that lived here. And I've been at a few parties where wild edible parties where people have prepared these, and you think they're going to taste like saltwater mussels. They do not taste good. You need to put a lot of like garlic and butter on them, maybe to taste sort of okay. So stick with the saltwater mussels, leave these freshwater mussels alone. My advice. All right, let's see. Oh, look at this picture. This is amazing with the sweet sign underneath it. It says, this tree frog seems to change color. When he's on the greenish beige siding, he matches that color closely. How does he do it? Asked Paul. So Brett, what's up with this sweet little frog that changes its color? Who is he and how can it do it?
First of all, I just love that they put the sign on the door handle. That's really great. This is a really good question. It's such a good question that I can't completely answer it. And my regular sources for information on amphibians, everybody agrees, and I've seen this myself, they do change color, and they can change color rather rapidly in minutes even, in order to better camouflage themselves. They also sort of have some change in color over their lifetime, in that the juveniles often are a bright green, and they tend more towards gray as they get older. But I couldn't find any information on exactly the mechanism by which this color change takes place. I don't know if John Benjamin, he may be able to answer that. I do want to say one thing though before I turn it over to him, which is that this is such an amazing trait of theirs, and a well-documented trait, that their scientific name, Hyla versicolor, is a reference to the fact that they have variable color and that they change color. So it's a distinguishing feature of this species, and it's really cool when you can watch it happen. And if you happen to find a gray tree frog, if you move it to a place with a slightly different background, you can sit there and watch it blend in better with whatever's behind it. So John, do you have more thoughts on the actual mechanism for the color change?
Well, a little bit, Brett. I'm certainly not an expert either, but from what I understand, there are two basic ways that, vertebrates can change their color in a way that's visible to our eyes. And one is slow and one is fast. So it's basically physiological versus morphological. So, in a simplified way, that is what I understand. Morphological is sort of like slow changing of how many pigment cells you have, on your surface. And, for reptiles like chameleons that can change really fast, it's a little more complicated because they have scales and a lot more stuff going on in their epidermal layer. But for amphibians, it's actually a pretty thin layer of cellular kind of situations. So, there's the physiological where they can actually just increase the number of cells, but that's a slow change. And the fast change, I think, is what we're talking about, how they can change before your very eyes, and that is a physiological change. So what I've read about it is, there's these cells called chromatophores that have melanin and other pigment molecules that are what we see as color, dark color. Generally, melanin is dark. There's other ones too, carotinin, xanthophyll, things we see in trees, and when they're changing color in the fall, that are universal throughout a lot of life forms. But the melanin is the dark stuff. That's what is changing human pigment color in skin, and it's very important thing among a lot of life forms. Basically, from what I've understood about it, they can change the concentration of the pigment granules in the cells from the center of the cell to the being dispersed throughout the cell. So when it's concentrated in the middle of the cell, then it looks lighter because you don't see all those dark colors spread out. And that can happen pretty quickly, apparently. I don't understand exactly how, but, the idea, I don't quite know what their cues are either for how they would know if they're on a, green surface or a dark surface. Like, what are their cues? But it's light, it's temperature, it's moisture, these things that they're picking up all the time. And they can change that in a visible way pretty quickly with just the way these cells are regulating and moving these pigment granules, like melanin and other kinds of dark, substances, molecules. So that's the best I understand it.
That's pretty cool. Thanks, John. Thanks, Brett. Those are great answers and such a great creatures. If I had another child, I really wanted to name her Hyla versicolor. Like her, that would be her first name, and her middle name would be versicolor. The gray tree frog has been my favorite frog since I moved here. Oh my gosh, this picture, what is it? This is was just so fascinating to watch was in the right place at the right time. What is this insect? What is it doing? What is the inflatable body part? Jenna, what the heck, man? What's going on?
So
we actually have a video as
oh
well.
yeah. Do you want to roll the video, Miles?
Yeah,
This is
awesome.
awesome. So I'm just going to talk while we watch the video. Is that okay? I'll be like our own little nature show.
Yeah.
So this is a giant ichnemon wasp, and she's female. What she's doing is she's pushing her ovipositor into the wood because she has detected with her nice long antenna that there is a larva inside the wood that she wants to lay her egg into. So what she's doing is she's got, it's hard to tell, but those sort of curly looking, those filaments they're almost like long muscle that she is using to help guide her ovipositor. So her ovipositor is her egg laying device. It's a few inches long. It's a hollow tube, and she will insert it into the wood, and she'll try very hard to hit the larva that's inside there, and then one little egg will come down her ovipositor into that larva. And then that egg will hatch out inside that larva and eat it, and then it will emerge eventually as another giant ichnemon after it develops inside the larva inside the tree. So this is one of my favorite insects of all time. I have several of these in my collection because people sometimes find them actually dead and stuck in trees because they do die this way. And they will like push that ovipositor in, and it apparently, according to something that I read this week after I saw this video, it takes about 40 minutes for the female to find the location and then successfully oviposit into the larva. And then she, of course, she has to do it again because she wants to have as many surviving offspring as possible. It finds the larva by smelling. So they will walk up and down trees using their antenna to smell. There was some suggestion that they sometimes can, once they've gotten closer with their sense of smell using their antenna, that they can also sense vibration of that larva that's inside the tree. But really, folks think it's mostly their sense of smell. Now, the crazy thing about these is that, so the male looks a lot like this, but doesn't have the ovipositor. The female has this long ovipositor and then these long structures that when she's not actually ovipositing or laying eggs, they look like one long tail, so to speak. And then if you can see that they're three separate sections, the center one being the ovipositor. And then there's this crazy enzyme, I mean, I think it's just crazy that this happens, that is exuded from the ovipositor as she puts it into the tree that helps sort of digest some of the wood so that she can slide her ovipositor more easily in. So there's all these layers that work. Then what we're referring to as the inflatable body part, this I've never seen before this video, that sort of membrane there. And in all the reading that I did after I watched this video, it seems that there's a membrane between several of her abdominal segments that helps, somehow it expands when she's ovipositing. I'm a little less clear on. So if anybody wants to do a little membrane research, go for it. But these, if you find a dead one, I would love it in my collection. It's one of my favorite insects to show children because they can't sting us. They are completely and totally safe for us. So if you see one of these, just watch her or him if it's a male. They don't sting with it. It's not strong enough it has no poison sac associated with it that could affect us in any way. So it's perfectly safe, although they look pretty scary when you see them because they're large. They're inches and inches long.
Wow, Jenna, that was just insane. That was like the insanest part of tonight. And we've had some pretty insane things from the porcupine necklace to the brainworm, but that was just crazy.
These are crazy. And there are smaller versions of ichnemon as well. So you may see smaller wasps that have a similarly, in proportion to their body, long ovipositor. they're all good to have out there because they're all going to lay their eggs into little larvae that are being pesty in some way. So let them all be. This one just happens to be giant. So
Yeah,
I am so grateful that we have a video of this. Oh, and by the way, doing my research about this membrane, I discovered that the very first photographic evidence of this particular species was in 1916. So we're over the 100-year mark. So this video is like, you know, I mean, we could say the first, I mean, I don't know if this is the first video of it, but this is amazing. It's a great video.
Wow. Yeah. Thank you, Pam. If you're out there, that was just such an amazing question. And we all learned something, which is one of the things we all love about Ask a Naturalist. All right, let's see what else is left to learn. Oh, how many tiny toads hopping all over the place here on Windy Row and Peterborough. They are too small to photograph, maybe just an inch in length. Where's the mother toad? How many of them will get to maturity? I've never seen this many in previous years. Why did the population surge? Signed, Chris. So Brett, tell us a little bit about the toad and its babies.
Yeah, so one thing that is different about amphibians, say, than birds or mammals is that most amphibians have very little parental care. There are some local salamander species that guard their eggs until they hatch, but most of the amphibians in our neck of the woods, the extent of their parental care is trying to pick a good place to lay their eggs, and then they wish them well and are on their way and have nothing whatsoever to do with the tadpoles or the toadlets after they metamorphose. So that mother toad is off in the woods eating bugs and living her best life and has nothing to do with any of her offspring other than picking a place to deposit the eggs and, of course, sharing her genes with them. I can't answer the question about how many will get to maturity. I did look for some studies on that. If you think about this, it's a very difficult question to answer because you would need to be following not just one clutch of eggs or grouping of eggs, but many, and then be able to follow those toadlets as they disperse through the woods. These toads reach sexual maturity. It can take, I think, a few years, if I remember correctly. So you'd really have to follow those animals for quite a long time over the course of their lives, and it would be pretty tricky to do that. So I can't really answer that part of the question. There are ways of tracking amphibians through putting tags on them and tags that have telemetry that can kind of transmit location information, but I'd imagine it's pretty tough with toadlets because as Chris mentioned, these newly metamorphosed toads are probably, you know, you can fit them on top of a penny. They're really tiny. The question about the population surge is a good one, and it's one that I get a lot around various species, you know. And I would say, yes, populations certainly, they fluctuate from year to year based on all sorts of things like, you know, weather conditions and predation and that sort of thing. But mostly what I see with regards to these toads is it's not that the population surge, it's that this is the first time Chris saw them. And maybe that's because they laid eggs near her house for the first time. They have some site fidelity, some attachment to breeding sites, but they're not exclusive. That's not exclusive. So we have toads at the Harris Center that sometimes will lay eggs in the little concrete frog pond, and some years they won't. Or maybe they'll go somewhere else. Maybe they'll use the pond at the bottom of the hill instead. So my guess is that it's not that the population surged, it's just that this year there were toad eggs that hatched near Chris's house, and so she saw them more. I see tiny toads hopping all throughout the woods every year in July and August, sometimes in great numbers. And when you see a whole lot of them at once, it's probably because you're very close to the body of water where their eggs were laid and where the tadpoles grew. And sometimes those bodies of water can be quite small for toads. They hatch in a matter of days, their eggs, and then they can take just 1 to 2 months to reach toadlet phase. So it doesn't have to be a big lake. And in fact, it probably won't be because they like shallow water. So it may be that she had, you know, a good wet spot in the woods near her house that some toads found this year. And that occasioned, gave her the occasion to see a bunch of the little ones dispersing from their breeding site. That's my answer. John may have something to add. I don't know. He's a herp guy too, so he's also into reptiles and amphibians.
I mean, I'll just share a story. I was running in Pisgah State Park recently, the Kilburn Pond Trail, which I love to run. It's a 6-mile loop down by the pond, and down the lower part of that, I saw tiny little toads that had just metamorphosed, hopping, and I realized there were just hundreds and hundreds of them. I mean, their little breeding zone had been right nearby, so I was literally stopped in my tracks so I wouldn't trample on these baby toads. I mean, they were just so abundant. I'd never seen anything like it quite that dense before, just how many of them there were. So, you know, I mean, the whole idea of it's a K selection, right, in biological terms, like lots of offspring.
Yeah.
Many of them won't survive, but they're all gonna go out there and do their best shot. And I was just trying not to step on them. I had the same experience with red efts sometimes around here. You know, this idea of just dispersing your offspring and making them, you know, get out in the big world and hopefully some of them will make it. So it's a cool thing to experience. I think it's a rare thing to see when they've just emerged from the pool and you can just see how numerous they are at that phase, you know.
And sometimes you can still see them with a little remnant of a tail from when they were tadpoles. And yeah, I imagine most of those end up being food for all sorts of things, turkeys and other birds. And Miles and I were walking on the Caddo Trail yesterday, and Miles heard a sound, and it was a wood frog still alive, being devoured by a garter snake. So I'm sure that many of these baby toads meet that fate as well throughout the course of.
I'll send you a photo, John. It was pretty awesome.
Amazing. Well, that's pretty cool. I just want to give say thank you. That was a great answer. And this is a little shout out for our friend Eric Masterson. I have Rusty here, his dog, and many of you know Eric had his hang glider had an accident with him on it, and Rusty's here even though Eric can't be. So here is a picture of an interesting mushroom that Peter and I encountered today. John, what is this varnishy looking?
Oh, wait,
mushroom?
this is a pole.
Oh, well, this is perfect.
I won't say anything yet, I guess.
Don't say anything. Mum's the word. Here's the poll. Is it a common stinkworm? Is it a shiitake? Is it a reishi? Is it a chick of the woods?
Chick of the woods is my vote. Chick.
Chick of the woods, baby. Wow.
Nobody thinks it's shiitake.
I love shiitake. And it's true. Andrea is making a comment about what a weird mushroom it is. It's a very strange looking mushroom.
All right, here we
All
go.
right, John, what is it? You want to tell us about it?
So among the options here, it is definitely a reishi, if I'm saying that right. It's a Ganoderma mushroom that is quite common, and I'm quite certain this is Ganoderma tsuge, often called the hemlock varnish shelf. So these are mushrooms that are, you know, they're shelf mushrooms. They're tough and woody. They can last for a long time. This is quite old in its life cycle, actually. This reishi here, I mean, it's already had its time to probably disperse most of its spores, and it's starting to turn dark red and kind of looking a little crumbly. So this is, as I think I said in a previous Ask a Naturalist session, this is a really valued medicinal mushroom. I mean, it's been for thousands of years in Asian cultures and also here in North America. I mean, I make tinctures out of it, which is super awesome. And there's a lot that could be said about that in even, you know, research that's been done on it as far as how it can help us immunologically, but also, you know, it's cool to know just the states that you want to gather them in. And this is not the one you'd want to gather. This is an old reishi, and it's you know, had its time, and you have to get them fresh, unfortunately, like a lot of mushrooms. But you can, I mean, Jeremy, this is clearly an old hemlock tree, right?
He signed off.
Oh, okay.
But yes, it is an old hemlock tree.
Old hemlock tree. So I mean, the two species that you'll find are Ganoderma tsuge, which is growing on old hemlocks, dead ones, and Ganoderma lucidum, which grows on old hardwoods. So, old oaks and maples. So where it's growing is, once again, very important to identification of the species. But they're both medicinal, both very cool species to pay attention to. And when they're young, they look so different. I mean, they're bright orange. And when you have the white little spore surface, that's when if you're interested in using them for medicine, you want to gather them when they're just fresh and sprouting from the base of mycelium.
So John, there was a question, you're using the word medicinal as opposed to edible. And so
Yeah.
there was a question about, is it edible? And you're saying not really, but it's medicinal.
Yeah, right. A very good question. Mushrooms harbor all kinds of compounds that can be valuable for us humans. And some of them are just good food, proteins and fats and all kinds of stuff. And you can cook them up and put them in your dinner. And this is not one of those mushrooms, because most shelf mushrooms are just too tough and bitter to really be tasty or healthy to just add to your dinner. But nevertheless, they just have all kinds of incredible polysaccharides, these molecules that have proven to be incredibly beneficial to us, both with folk cultures for thousands of years, and even modern studies, that are kind of getting to the scientific base of why they're helpful. But I mean, for my part, I just love to just consider them free medicine from the forest. And there's certainly nothing that I've experienced that's dangerous. And so what I do is, for these kinds of mushrooms, I make what's called a double extraction tincture, where you can soak them in alcohol for a while, which gets all these long molecules out of the mushroom that are very complex, and you couldn't just get them out from boiling it for tea, because they're too delicate. And then you do that for like a month or so. And then for the rest of the mushroom, you boil it, and you get the sort of smaller molecules out that can be extracted through just boiling water. And you mix them together at the end of the day, and you have this tincture. And so if you want to learn more about it, you can send me an email. But I will.
Great. We do need to move on. We're already a little over, because this is such a juicy Ask a Naturalist. But here's our next one. I was kayaking at Nubanuset on Monday, and I found what I believe to be 2 baby bald eagles just hanging out on shore. So cool. They allowed me to sit and observe for a while, and then eventually each showed off its flying skills by flying to low branches nearby. What an amazing experience. I was wondering if you know how recently they fledged. I'm thinking pretty recently. Brett.
I can answer this one. I've been kind of I know several of us, including Susie and Jeremy, have been watching these eagles over the course of the summer. And I was in touch with Chris Martin, who is the New Hampshire Audubon's raptor biologist, who kind of oversees bald eagle monitoring throughout the state. And so I was in touch with him about this particular eagle pair several times this summer, or the pair meaning the parents of these 2 young birds. And so Kim just commented that she saw them on July 20th, which I can tell you that the first chick fledged somewhere between July 5th, when I saw both of them in the nest, and July 10th, when Jenna's husband and sons saw one of them on the beach being fed by its parents, and the other one still in the nest. And so the second one fledged sometime after that, probably within a few days of each other. These chicks typically fledge after 11 to 13 weeks in the nest. So they're up there for quite some time. And this particular nest has a really long and storied history. I won't go into all the details of it, but it's a really interesting spot. This was the second occupied bald eagle territory in all of New Hampshire after DDT was banned. So after bald eagles nearly disappeared because of DDT, Susie actually found this nest. What was it, in the early 90s, Susie?
Yeah, probably like 92, 93 maybe. Yeah, it was so exciting.
Yeah, and at that time it was only the second known bald eagle nest in the entire state. Now there are many, many more. And this one in most recent years has had some challenges. This pair who's there now, they're both known eagles. They're both banded. They were both banded as nestlings when they themselves were nestlings. So the female was banded in 2008 near the Mass Pike. And the male is actually the oldest wild bald eagle known in New Hampshire. He was banded in the spring of 1997 in the Quabbin Reservoir. So he's a 23-year-old bird now. And they've been nesting together at Nubanusset for a number of years, but the last few years they had some really bad luck and also probably some bad nest building. Their nest collapsed several times, which led to the death of their chicks for a couple of years in a row. And then last year, I think it was last year, it might have been 2 years ago, the tree that their nest was in fell over completely, which also unfortunately led to the demise of their young that year. So this year is really exciting. It's the first year in probably 4 or 5 years that chicks have been successfully fledged at this site. And the fact that there are 2 is even better. So it's a really good turn of events for this eagle family here, and that they're still there. That Susie was just saying she saw them, was it over this past weekend?
Yeah, this Monday I was out with my boys, and they were fishing, and they had caught a fish and they let it go, and just after they put it back in, we had been hearing the baby eagles kind of begging. They put the fish in the water, and the adult eagle came and swooped down and got the fish and flew off, and then the babies came out and followed them, and then I think the mother fed, or the adult fed the babies the fish, because we didn't hear them begging for food anymore. But one thing we noticed is the young ones are very clumsy flyers, and if you ever see one of these guys up in the air next to the adult, they're actually bigger than the adult. Their wingspan is bigger, and that's because they aren't so good at flying. Their wings, their feathers are larger to help them compensate and be able to kind of fly a little bit better. And then eventually, as they age out, which takes them about 3 or 4 years, their feathers and size will come into the same kind of size as the adults. It's so cool. So you can still see him on Nubanusset, up to shenanigans.
And New Hampshire isn't banding nestlings as much as they once were, but Massachusetts is still making an effort to ban every bald eagle nestling in their state. So, and a lot of those birds eventually end up here. So it's a really great story that bird banding can tell us. So
wow, we are really good. We went over. We hope you don't mind. It was really a juicy evening, and I want to just encourage everybody to keep sending their questions. Our next Ask the Naturalist is September 24th, 5:30 to 6:30, right here on Zoom. So send us your questions, and in the meantime, get outside and explore. And have a great evening, everybody. Thank you especially to our panelists, and Mark, who had to leave a little early. If he was here, we'd give him a big round of applause. So thank you all.